Bismillah ar-Rahman ar-Rahim. We start with the last chapter, which is genetic technology. This is the first video of this chapter, which is a very complex and difficult chapter for all of us. We're going to talk about basically recombinant DNA technology, and we are going to discuss it in a number of ways to make you clear on this topic, which is quite challenging. First of all, I would like to introduce you to a few terms which we are going to use now. One word that we use is a palindrome. And I'm sure you might have heard of it or you might not have heard of it. Is a word or a phrase or a verse or a sentence that reads the same forward or backward. Now, if you can look at these, civic, C-I-V-I-C, well, you can spell it this way or you can spell it this way. It means the same thing. Race car, R-A-C-E-C-A-R. So it's both ways. This ways or that ways. It's the same thing. Radar, refer, reviver, rotator. So palindromes, madam. Of course, your favorite is LOL, which can be read this or it can be read that way. It's the same thing. So mom, dad. So all these words you can read both ways, and this is called a palindrome. Now we're going to be talking a lot about this in as we go ahead of this, how restriction enzymes cut at palindromic sites. So I want you to know these words, and I want you to be very familiar when I use this word palindrome. Now, the first thing to do, as usual with every chapter, is we go through the syllabus. And the syllabus has a number of points, which is the basic one is the define the term recombinant DNA, and then of course the different points as we go along: polymerase chain reaction, gel electrophoresis, why do we use plasmids, why do we use promoters, why do we use fluorescent markers, what is the role of restriction enzymes, reverse transcriptase and ligase, and the last is what is microarray. So I will be handling these maybe not in the same sequence, but maybe in a different sequence. But I will try to cover each of these topics, and then we will do a recap at the end of all these videos. We have 19.2, and these are further subdivisions of the syllabus. Genetic technology applied to medicine, in which we have to study what is bioinformatics and the advantages of genetic technology and the genes BRCA1, BRCA2, genetic diseases, social and ethical considerations, PCR and DNA in forensic medicine. Then 19.3 is genetically modified organisms in agriculture. So the importance of BT maize, vitamin A golden rice, and GM salmon. All these, of course, we will discuss in how we genetically modified organisms in the agriculture industry and how it is beneficial to us to get higher yielding crops or better crops in some way. So all these, of course, there are a large number of people like myself who are very much against genetically modified organisms, and we do not agree with this, that because it has far-reaching effects, and people who eat this food might have adverse effects, and we might learn 10 years from now or 15 years from now. So what are the ethical reasons why some people do not like it, and some people say no, it's okay, we need to do these new maneuvers to increase crop yields. Then I want to discuss reverse transcription. Now, you know what is transcription. You see, I mean, if you remember from your AS level, transcription was DNA to mRNA. Now, here we are doing something which is the opposite of this. We are doing mRNA to DNA. Now, why are we doing this? We are doing it for a certain reason, because we can find mRNA. Like, for instance, if I need the mRNA to make insulin, well, I just have to go to the beta cells of the pancreas and I take out the mRNA. So reverse transcription means that we take the mRNA, we give it an enzyme called reverse transcriptase, which we get from a virus. The AIDS virus has this enzyme. And what do we get is we get cDNA. And the mRNA is removed, and the second strand of DNA is made by using another enzyme called DNA polymerase. So we get the mRNA, and then we get a single-stranded DNA, then we use DNA polymerase, and we get a double-stranded DNA molecule. So what we need to understand is that why are we doing this? Why are we doing this is because we need the gene for insulin. Now it's very difficult to find the gene for a certain protein, But if we know where it's being made, like we know that insulin is made in the beta cells of the pancreas. So if it's in the beta cells of the pancreas, well, what we need is from the beta cells of the pancreas, we need the mRNA. So we get the mRNA out of that. And from the mRNA, we can make the DNA. But please understand, this DNA is not the normal DNA. We call it cDNA. And why do we call it cDNA? That is what we need to understand. And I'm just going to explain this as we go along. Then we need an enzyme called DNA polymerase. Now, what does the DNA polymerase do? It's an enzyme that synthesizes DNA molecules from deoxyribonucleotides, which are the building blocks of DNA. And we need this in DNA replication, and when we need it to make two identical DNA strands from a single original DNA molecule. So DNA polymerase, we need it because we need to make, if we have a single stranded DNA, like for instance, I just told you we had the mRNA. So from the mRNA, say this mRNA is this. From the mRNA, we get a DNA, and then we need a double-stranded DNA. So we need the enzyme DNA polymerase. So this would be a double-stranded DNA made from an mRNA. Then we need an enzyme called DNA ligase. And what does it do? It forms a phosphodiester bond, linkage between the double strands. And when we have to connect a different DNA with the vector, which is the plasmid. So DNA is the enzyme used for joining the cut strands of DNA together. Of course, uh, it is going to form, remember, the phosphodiester bond. And the phosphodiester bond is the one which we are going to be reforming, because we might have a plasmid and we have another different DNA, which is human DNA, but then we need to join it up. And we need to join it up, and we need an enzyme called DNA ligase to join it up. So DNA ligase. Now we use this word a lot, and so I want you to be familiar with this, that DNA ligase seals the nicks. Nick is the gap, is the phosphodiester bond which needs to be uh, constituted, it needs to be put together. So DNA ligase seals the nicks between the Okazaki fragments, which are of course fragments which are made in DNA replication. It closes the nicks in the double-stranded DNA. So this is just a word that we use, and I want you to be familiar with this word as well, which we keep on using a lot. Now, the next heading or the next topic which I want you to understand is what are restriction enzymes. Now, if we understand this, we can easily understand the whole chapter, which is on genetic technology. First of all, you've got to understand is that these are only found in bacteria. And these were present in nature, but the person who discovered them and he got the Nobel Science Prize for that. Restriction enzymes are present in bacteria. Why? Because you have to understand that bacteria has to protect itself. So, say there is a virus which is attacking this bacteria. Now, when the virus attacks this bacteria, what you have to understand is it injects its DNA. So, the DNA of this is a virus, and this virus has a DNA, and it injects the DNA into this bacteria. But what does the bacteria have? The bacteria has an enzyme. The bacteria has an enzyme. Please understand this. The bacteria has an enzyme which is going to recognize that this is not my own DNA, this is alien DNA, and it is going to cut it up into pieces. So, bacteria is very clever. Bacteria is very smart. What it does is it has enzymes. God has given it enzymes, like we've been given lymphocytes, which make antibodies to protect us from pathogens. Now, what the bacteria has is restriction enzymes. Bacteria has restriction enzymes which can recognize and cut alien DNA. Please understand, alien word, not to your own. So, now what do we say? Restriction enzymes, bacterial enzymes, they cut DNA molecules only at restriction sites. And these are categorized into two groups based on the type of cut. Either they will result in sticky ends or they will result in blunt ends. Now I'm going to explain it to you what is sticky ends and what are blunt ends. Now if you look at this double-stranded DNA, this is a palindrome. Do you see how this is a palindrome? GAATTC this way and GAATTC this way. Now, what does a restriction enzyme do? It cuts between GAATTC. GAAT doesn't cut between just G and A, it cuts between GAATTC. So, what sort of an enzyme is going to do that? The enzyme is going to something look like this. It's going to fit in here, it's going to have an active site where all these 6 pairs of DNA fit. Now, this restriction enzyme is specific. It doesn't cut just G and A, please understand that. The active site has all these 6 base pairs fitting in there and cutting it between G and A. And you get these. So, what will you get? You get this piece of DNA and you get this piece of DNA. So, this piece of DNA separates. And then I'm using another color to show this piece of DNA and this piece of DNA separates. And what do you get? You get sticky ends. Sometimes, of course, it cuts straight in the middle, so you get blunt ends. So, blunt ends and sticky ends. So, restriction enzymes are specific enzymes which are found inside bacteria and they recognize and cut alien DNA. But that is what we discovered. So, what we did is we got these restriction enzymes and now we use them in genetic technology to cut DNA of our own choice. So now what do we have? What are restriction enzymes? Basically, I called them a DNA scissor. They are a DNA scissor, like you have a scissor for cutting your hair, you have a paper cutting scissor, you have a scissor for cutting if you're making a shirt for yourself or cutting a cloth for yourself. So restriction enzymes are basically a tool for genetic engineers to cut DNA at specific sequences. This is an example of blunt ends versus sticky ends. You can see how this one is different, how this results in sticky ends, and how this results in blunt ends. So the names of these restriction enzymes are very specific, like EcoR1, because it was obtained from a bacteria called E. coli, and we called it restriction enzyme 1. There's EcoR2 and like this. And this one, of course, was discovered from some other bacteria and we call it SMA1, and that is how it cuts. So I want you to pause the video here. I want you to understand what do I mean by blunt ends and what do I mean by sticky ends. If you understand the basics, then we can go on to further understanding the entire chapter. It will become very easy for you. Now, an overview of genetic technologies. Number one, first of all, we take a human pancreatic cell. We extract the insulin gene. Similarly, we take a bacterial cell and we extract a plasmid. So this is the bacteria, this is the plasmid. I somehow break up the bacteria and I obtain the plasmid. Now, introduction of insulin gene into the bacterium plasmid. So first of all, I have to cut this plasmid and then I have to find the human, from the human cell I have to identify the insulin gene, which I have to cut because this must be in part of a chromosome. So I have to cut this out, and then I've got this human insulin gene, and then I put this human insulin gene here, and then I use the enzyme ligase to glue it together. Now what do I have here? I have recombinant DNA. I have recombinant DNA here. So what did I first do? From the human cell, I got the insulin gene. From the bacterial cell, I got the plasmid. I cut the plasmid. I cut the human gene. Then the introduction of the insulin gene into the bacterial plasmid. Now I have to get this recombinant DNA into another bacteria. Those bacteria are gone, the ones which I recovered the plasmids from. Then inserting the recombinant plasmid into a bacteria, and then we grow these bacteria, and these bacteria produce insulin for us. So the basic overview of recombinant DNA technology is the gene of your choice. Gene of choice, like for instance, I need insulin to be giving it to patients who suffer from diabetes. So that gene, then we take a bacteria, take out the plasmid, cut the plasmid, take out the human gene or any other gene that we need, we put that and combine it. So plasmid plus the human DNA, plasmid is also made of DNA, but that's bacterial DNA. So basically we have got the bacterial DNA, this is the bacterial DNA, and then we've got the human DNA, and the red is the human DNA, and then we've got recombinant DNA, and then we put it into another bacteria, and then we grow these bacteria in a fermenter, and they're going to produce insulin for us. Now, this is an overview of gene transfer, and I'm using the prescribed book, which has been told to us to be uh, used in A-level biology. Now, the 5 steps that we talk about are very simple. The first is the gene that is required is identified. First, we have to find out the gene for insulin. It may be cut from a chromosome, or it may be made from mRNA using reverse transcriptase. So either we cut it from the chromosome or we make it from the mRNA and then we get the DNA out of it. Or we synthesize it from nucleotides. We can even use nucleotides to synthesize this gene. Then multiple copies of the gene are made by using a mechanism which is called PCR, which is called polymerase chain reaction. We'll talk about that later on. Then this gene is inserted into a vector. Now the vector can be anything. It can be plasmids, it can be viruses, it can be liposomes. What are the 3 vectors? Number 1, plasmids. Number 2, viruses. And number 3, liposomes. Liposomes are just little circles of phospholipid bilayer. And these are called liposomes. And we put the DNA of our choice into it. And we may give it in the form of an inhaler, or we may give it in some other way, so that it goes and reaches the cells where we want this DNA to undergo transcription and translation, and that normal protein should be made, which was not being made previously. Then the vector takes the gene into the cells, and the cells that have the new gene are identified, and then we are cloned, like the bacteria that we used. And then, so these are the steps of how the recombinant DNA technology is going to be talked about. For these steps, the genetic engineer will need something, and then I've talked to you about what we need. We need restriction endonucleases, which are also called restriction enzymes, or endonucleases, same thing. Then we need the enzyme ligase to form the phosphodiester bond, and we need the reverse transcriptase to form, maybe if we are using the RNA, from the RNA to make the DNA. The vectors including plasmids and viruses, and I've just said liposomes. And then genes coding for easily identified substances can be used as markers. The reason is that, you see, we don't know which bacteria contains the recombinant DNA. Is it this bacteria, or is it this bacteria, or is it this bacteria? So in order to identify them, we need to put some sort of a marker along with the gene that we have introduced. So we might put some sort of a marker here, so that then I can identify that this is the bacteria that I am going to culture, not these, not these. Because a very small percentage of bacteria actually take up the recombinant DNA. We are not very successful. Say if I have 100 million bacteria and I mix them with the recombinant DNA, well, only a very small percent, 20% will take up the required recombinant DNA. So I don't know which ones are those. So I need to identify them by using some sort of a marker gene, which I have then introduced along with the gene which I have introduced into the recombinant DNA. Now, what is the definition of recombinant DNA? That's the first part of the syllabus. Production of a unique DNA molecule by joining together 2 or more DNA fragments not normally associated with each other. And the DNA fragments are usually derived from different biological sources. I just talked about it. So we've got the plasmid, which is bacterial DNA, and then we've got the human DNA. Now, this is a recombinant DNA because part of it is bacterial And they're two different organisms, two totally different organisms. And this is the bacterial and this is the human DNA. So this is called recombinant DNA. This is a new combination of the DNA. So please understand what is recombinant DNA. Then an overview of it. Why are bacteria used in recombinant DNA technology? Number one, bacteria easy to grow, maintain. They're very easy to grow. They can be maintained and manipulated in laboratory, multiply very rapidly. Bacterial plasmids can be manipulated, and cloned recombinant plasmids can be readily isolated from bacteria. So basically, why bacteria? But the main reason is, of course, that they multiply very rapidly, and of course, you can manipulate them in the lab and you can easily work on them. And so we need something which is easily workable. Now, this is a very good overview of recombinant DNA technology. Of course, we go into more details as we go on. You can see here, this is the plasmid, and this is the foreign DNA. And then we have cut the DNA using an enzyme EcoR1 endonuclease. And you see how we have now got the sticky ends. And then we have mixed the cut plasmid and we mix the foreign DNA. And then what do we need? We need an enzyme ligase. And then we've got this plasmid which has become a carrier. So plasmid, the one in red, the blue is the foreign DNA. We have used the same restriction enzyme to cut the plasmid and the foreign DNA so that we get the same sticky ends. And then what we've done is we mix the plasmid and the foreign DNA and we get half of it is the plasmid, the red, and the blue is the foreign DNA. And then we've used the enzyme DNA ligase, and now we have a plasmid chimera or a recombinant DNA. And you see how very easily it joins up because the sticky ends are very easy to match up. The sticky ends are the same on this side and the sticky ends are the same on this side. So you need to be very clear how the sticky ends are a great help. And the sticky ends are going to be very easy. So hydrogen bonds will form between the different base pairs. A will pair with T and C will pair with G. So we get a very quick mixing of it and the base pairing will occur automatically. You don't need any enzyme for that to take place. Now, another word that I use very often and I talk about in genetic technology is what is a bacteriophage. And you immediately think of it as, oh my God, there's some sort of a new monster bacteria. No, it's not a monster bacteria, it's actually a virus. And we usually just call it a phage. And that's the nickname of it. So, phage and bacteriophage is a virus that infects and replicates within bacteria and archaea. So, if we look at it, and I've just drew this diagram, this is a virus, and this has got a DNA. Now, this virus, which is going to attack a bacteria, this virus which is going to attack a bacteria, so is called a bacteriophage. It's a virus, but it's called a bacteriophage. So, bacteriophage are viruses that live on bacteria, that infect bacteria. You must realize that all viruses have an affinity for different organs of the human body as well. Like, for instance, there is the hepatitis virus, and everybody knows that hepatic artery, hepatic vein. So, the hepatitis virus is going to infect your liver cells. So, the AIDS virus infects your helper T lymphocytes. So every virus has its affinity or likes certain organs of the human body if they infect humans, and every virus which has an affinity for bacteria and attacks bacteria is called a phage. It's a virus. So bacteriophage, you must be very clear about it. It's not a bacteria, it's actually a virus that infects and replicates within bacteria and archaea. So bacteriophage. I want to talk about the biological role of restriction enzymes, and you wonder why do we have restriction enzymes in nature? Why did God give those to us? Well, because restriction enzymes are paired with methylases. Methylases are enzymes that add methyl groups to specific nucleotides within the recognition sequence. The methylation prevents recognition by the restriction enzyme. Now, the restriction enzyme in a cell, like in a bacteria, doesn't destroy its own DNA. However, the restriction enzyme can destroy foreign DNA, as I mentioned earlier, alien DNA. So the role of restriction enzymes is actually protection. The bacteria has its own protective system, has its own immune system, and that immune system is its restriction enzymes. And those restriction enzymes will cut alien DNA or foreign DNA, but it will recognize its own DNA and will not cut its own DNA. Why? Because its own DNA is being methylated. So the methylation prevents it being cut by the restriction enzymes. Now here's another picture just to make you understand it. Maybe I've not been able to clarify it earlier. You see the bacteriophage. As I told you, a bacteriophage is a virus. Now the bacteriophage, the virus injects its DNA, and you can see the bacteriophage DNA. And then you see the bacterial chromosome. And you see the restriction enzyme EcoR1. Now the restriction enzyme EcoR1 is the property of the E. coli bacterium. And look what it does. What it does is it fragments the bacteriophage DNA. So it just breaks up the bacteriophage, the viral DNA, which is the bacteriophage DNA, is just broken up into tiny fragments, is broken up into pieces. So the poor virus cannot replicate anymore. The bacteriophage slash virus cannot replicate anymore. So restriction enzyme act as immune system in the bacterium. When a bacteriophage, a virus, tries to inject its DNA into the bacteria, restriction enzyme cut up the bacteriophage DNA into many fragments, thus preventing it from doing any harm to the bacterium. Well, that is what we need to do research on and find out how we could use it to cure viral diseases. But nobody's thinking on those lines, and all we are thinking about is vaccines. We're not thinking of how to really treat viral diseases. So this is one of the main areas of research which can be followed by people who are doing research on to treatment of viral diseases. Now that finishes this first video on genetic technology, and I hope to see you in the next video as well, which will follow soon. Thank you.